This is the review video for Lecture 7 Part 3, which covers things like antigen presentation and more details about the immune response, and gets into how does the adaptive immune system work and why are all the lymphocytes so weird the way they are. So by the time you do this review, you should know what an antibody is and how it works. And you should know what a lymphocyte is, but there's a lot of other things we need to get into. So the first thing I might want to review is lymphocyte education. You might have gone over this in part 2 and you might not, but lymphocyte education involves 2 tests. It's where the lymphocytes are tested for do they function, and then do they recognize any human protein or self-antigen? So it's always useful to go back and look at a lymphocyte whenever you're talking about lymphocytes. So let's draw a lymphocyte, and this ought to be great. Should be a circle. That's what they look like under the microscope, and if you want to fill in the nucleus as it would appear in typical histology, it's going to be a bluish purple blob that fills up most of the lymphocyte. So that's what a lymphocyte's going to look like. Yeah, that's what a lymphocyte's going to look like. That's what it looks like under the microscope, but that kind of doesn't show you the most important parts of the lymphocyte. So I want to draw some B cells and I want to draw some T cells. Just make sure we are all on the same page. So I'm gonna try to make this work. Okay, now we're looking at a bunch more lymphocytes and we can draw their receptors. So I'm gonna make these 2 B cells and these 2 down here T cells. And if you're reviewing with me, you should be drawing this faster than I am, right? You should draw some B cells and T cells before I do, or stop the video. Just make sure you're going to do what I do. And be very careful when you draw a B cell receptor, because it's a Y within a Y. B cell receptor is basically an antibody, but instead of the Fc portion, it has a membrane portion that can signal the rest of the cell if it binds something. And I'm always going to draw a B cell receptor with some kind of shape for its binding sites. And that shape, that binding site, is what is randomly generated by shuffling DNA. So each of these lymphocytes has a gene or a set of genes that encode the binding site, and those genes were randomly generated. The rest of the receptor is the same from B cell to B cell, but the binding site is not. And so here's where we need the key terminology. I keep saying, does it recognize? Does it bind? Does it attach? And this is where you have to understand what a receptor is. Go back and look it up if you don't know. But a receptor is something, it's a protein that has a very specific shape, and one thing can bind to it. One thing fits it perfectly. And what we say is if that thing fits perfectly into the receptor's binding site, we say receptor has bound it. So if the receptor can bind it and it touches it, it binds it. It has bound it. But at the same time, a receptor isn't just meant to stick to something, it's meant to signal the cell that it has stuck to something. So we can also say that a receptor recognizes something, because when it binds to it, it knows that thing it bound to is the one thing it can bind to. So it has recognized it. Now with lymphocytes, they don't know what they're looking for, but they have a very, very specific binding site that will only recognize or bind to one protein, more or less. So that's what you should think of when you look at these binding sites, is what shape would work with those binding sites, and that's randomly generated. Now T cells, we're always going to draw with a different shape receptor, because T cell receptors are different, and you should be able to tell me the difference. What is the big difference between T cell receptors and B cell receptors? And the main difference is that T cell receptors need another cell to present an antigen to them. They can't just look at an antigen. They can't bind to anything that isn't being presented to them. And they generally need co-receptors to make it work. But they're all going to look the same under the microscope. The only thing that's going to be different between them are the types of co-receptors they have and, of course, the randomly generated binding site that they happen to have. Okay, so that was long-winded, but when people don't know these things, the rest of it can't make sense to them. So we have to review this. Always have to review this. So what we did was I mentioned lymphocyte education, and then I review lymphocytes. So you saw B cells and T cells. So that's important. Okay, so the next thing is, what question is a lymphocyte asking? And we go right back to our lymphocytes, and there's only one question they can ask, which is, does this protein fit in the binding site of my B cell receptor or T cell receptor? Do I recognize this thing? Is this the thing I'm looking for? That is always the question that they are trying to answer. Now, in different contexts, the answer to that question will lead to a different part of the immune system. So we have to remember both what is the general question they're always answering, and in different contexts, what happens if that answer is yes. So our next topic is antigen presentation. Antigen presentation is how T cells look at antigens. So let's draw it out and use some color. So with antigen presentation, we're always going to have some kind of cell that presents an antigen. So this is just a cell. It could be lots of different things. And then the other partner here is a T cell, because the antigen is always being presented to a T cell. So the parts that we know, that you certainly know, you can always draw, include the T cell receptor. And that receptor is trying to bind to, or hoping it will bind to, the antigen that is being presented. And that antigen is being presented on MHC from the other cell. So the other cell presents that antigen using its MHC, and the T cell asks, does that antigen fit the binding site of the T cell receptor? The other part here is the co-receptor. All right, so now let's label everything, and let's label it well. So what do we have? We have a T cell receptor. We have MHC. We have an antigen or epitope. It's better to call it an epitope, but nobody does. And we have a co-receptor. Now, the devil is in the details here. So, we have two types of MHC and two co-receptors that we talk about. So that's something we need to talk about. So in antigen presentation, we have types of MHC and co-receptors. And you have to know a bit about the MHC to understand what's going on. So what we have is MHC1 is in all nucleated cells. By in, I mean they are all capable of using it, and they use it to defend the body from whatever might be infecting them. And MHC1 presents cytoplasmic proteins. All right, so MHC1 is for presenting cytoplasmic proteins, and it fits co-receptor CDK. 8. So MHC1. MHC2 is in macrophages and dendritic cells and B cells and some others. It presents proteins. Well, technically it presents endosomal proteins. That means proteins from endosomes. Endosomes are the vesicles that result from endocytosis. So generally speaking, if a cell does endocytosis, it brings in something from the outside, and that thing ends up in a vesicle, and that vesicle is called an endosome. Normally, we're talking about phagocytosis, and phagocytosis brings in something large, and that's inside what we call a phagosome. It's just a type of endosome. So, always with MHC2, a cell is displaying something it brought in from the outside, whereas with MHC1, it's displaying something it made on the inside, with one exception. And MHC2 fits CD4. So that's what you need to know about the MHCs and the CD8 and the CD4. So the next thing we looked at is cytotoxic T cells. So let's draw a cytotoxic T cell. It looks like this. Now a cytotoxic T cell is CD8 positive. So let's just real quick draw a cytotoxic T cell. Okay, so it's going to look like this, and it's going to be CD8 positive cytotoxic. And it's going to look with its T cell receptor at antigens presented to it by any nucleated cell. Any cell, basically any cell, is going to display some antigen. And the cytotoxic T cell is going to look at that MHC. It's going to align it using CD8 coreceptor, and that is MHC1. Okay, so then the question is, what do cytotoxic T cells do? Well, you can look it up and it'll say they kill cells that are infected by viruses, or they kill cells that are cancerous, or they kill abnormal cells. Well, how do they know? How do they know what cell is abnormal when all they have is a T cell receptor? And the answer to this is it's not rhetorical. They don't know. They don't know anything except that they kill any cell that presents a protein they recognize. So, go back and look at lymphocyte education if that didn't make sense to you, because there is perfect logic to it. Look at lymphocyte development, look at lymphocyte education, and understand this, or ask people questions, because this is fundamental to how the immune system works. We just have a huge pool of cytotoxic T cells, huge pool of mature, naive CD8 positive T lymphocytes that could be cytotoxic T cells. And each one of them is only capable of killing a cell that shows it that one random protein that it happens to recognize. So that's a cytotoxic T cell. But then, what if the cell, what if the little virus inside the cell takes it over and it doesn't display MHC1? That is a trick viruses will use. They will not show MHC1. And that means a cytotoxic T cell won't see the virus. There's no way it can see inside the cell, because without MHC1, this cell cannot see internal proteins. That's what MHC1 is for. So, cytotoxic T cells cannot hurt cells unless those cells have MHC1. So this virus is getting away from the immune system. This virus is winning by forcing this cell to stop showing MHC1. And that is where natural killer cells, which I'll abbreviate NK cells, natural killer cells recognize this lack of MHC, and they will kill that cell. So natural killer cells are part of the innate immune system. They don't look for some random thing. They're looking for an absence of MHC1. And they're born with all the receptors they need to detect that. And they're some of the first cells that help you during a viral infection, because most viruses do this. Most viruses tell the cell to stop making MHC1. Okay, so that is cytotoxic T cells and natural killer cells. Make sure you understand how they work together. Okay, B cell education, and it has several steps. One thing to understand is that there are cells that can present antigens to B cells. So, but they don't do it using MHC. I'm not sure how they do it. So B cells can both bump into antigens and free, and they can find antigens that are being displayed by macrophages and dendritic cells. So here's how it works. Mature, naive B cells circulate in the blood and lymph. And then they come to rest in lymph node. And this is where the antigen-presenting cells like macrophages and dendritic cells will be hanging out. Macrophage or dendritic cell show antigens. And if a mature, naive B cell recognizes an antigen, if, it's a big if because it almost never happens, if it recognizes an antigen, it starts to make immunoglobulin M. That's the first antibody that appears during an immune response, but it's not the best antibody. And it doesn't make huge quantities of it, and it doesn't go through clonal expansion or differentiation. What it does instead is it internalizes its receptors. So its receptors are what bound to the antigen. It brings them in through something like phagocytosis and digests them and grabs the protein antigen that they recognize, and it presents antigens to helper T cells via MHC2. So you get that? A B cell recognizes and binds its antigen when another cell shows it to it, and it brings it in and then gets ready to display it on MHC2 to helper T cell. If the helper T cell recognizes the antigen, will give the activation signal. B cell will go through clonal expansion and differentiation, and you'll get plasma B cells and memory B cells. But what did it take to do that? What did it take to go through B cell education? It required a B cell that recognizes whatever antigen is in the body, finding that antigen, and then showing it to a helper T cell that also recognizes the exact same antigen. And both of those are rare. Both of those are like 1 in a million cells. And so this is only ever going to happen if we've got tons of an antigen in the body, like we have a virus infecting the body, filling it with spike proteins and capsid proteins. So B cell education is a two-step process, and what you'd see is a few days into a viral infection, you'd see immunoglobulin M, and then a few days later, the plasma B cells would start making whatever antibody is appropriate. So during viral infection, they'd make IgG. During a worm infection, they'd make IgE, and that's B cell education. Okay. The next topic is antigen presentation situations. So these are the scenarios that you would need to understand. So what we're going to write out is a cell uses MHC, which MHC, to ask another cell, to ask which cell, a question. So let's get into that. So a dendritic cell uses MHC2 to ask mature, naive, CD4 positive T cell, should I activate? Okay, that's, oops, what did I do? Not MHC11, MHC2. Should I activate? So a dendritic cell is helping a helper T cell figure out whether to activate or not. So it finds mature naive T cells and shows them all the antigens it's been able to collect, and hoping that one of those mature naive cells will actually recognize an antigen. And again, T cell education has killed all the T cells that might recognize a human protein. Most of the proteins in a human body are human proteins unless we're being attacked. So the only time we're going to activate a helper T cell usually is when we're being attacked, and we have some foreign thing that some helper T cell will actually recognize. So that's how we activate a helper T cell. A B cell can use MHC2 to ask a CD4 positive helper T cell, should I activate? I screwed this up. Where is the error in what I put up here? It's here. The question that that a dendritic cell asks a T cell is, should you activate? Sorry, I got that wrong. So, but the question a B cell asks a helper T cell is, should I activate? A macrophage can ask, whoa, hey, no, not that. Can use MHC2 to ask a helper T cell, should I become more aggressive? A dendritic cell can ask a helper T cell, may I be licensed? So may I start activating CD8 positive T cells? Those are the major MHC2 situations. The first one is activating a CD4 positive T cell. So activating a helper T cell. It starts as mature naive, but once it sees its antigen, it gets the signals and it goes through activation. So then you have the helper T cell, and there are a ton of different situations where cells will show a helper T cell an antigen and say, hey, do you recognize this? And ask it a question. If you recognize it, I will activate. If you recognize it, I will become more aggressive. If you recognize it, I will be licensed. Because in all cases, they're saying, I found this protein. Is this important enough that there's a helper T cell looking for it? So then that's MHC2. And again, MHC2 is for proteins that are brought in from the outside. MHC1 is for proteins that are made inside the cell. So a licensed dendritic cell can ask, can use MHC1 to ask a CD8 positive mature naive T cell, should you activate? So this is activating a cytotoxic T cell. It's finding the mature, naive, CD8 positive T cell that has the right receptor. And then once we have an activated CD8 positive cytotoxic T cell, we can start seeing any nucleated cell has to use MHC1 to ask a CD8 positive cytotoxic T cell, should you kill me? Okay. That is the antigen presentation situations. Okay, so what's left? One short topic, and then I'll tell you about two topics that we skip. The short topic is the complement system. Look at how that's spelled, and look up complement, as in addition to thing, versus compliment as a nice thing you say to someone. They're not spelled the same way. But it only matters if you care. I'm not going to mark you wrong for misspelling complement. What you need to know about the complement system is, what is it? It is a system of proteins in blood. It's proteins in blood that have 3 functions and can be activated 3 ways. So make sure you have nailed down and reviewed the 3 functions of the complement system and the 3 ways it can be triggered. 2 of the ways it can be triggered are triggered by the innate immune system, basically doing innate things that we don't need to wait for an adaptive immune response to do. And then the other way it can be triggered is by antibodies, and those might not appear till several days into an immune response. So the complement system can protect our blood from bacteria pretty well, and it's even better at it if there's an immune response going on targeting those bacteria. That's all I'm going to say about the complement system. And that's it. We didn't look at anything else in lecture. There isn't time, but what I wanted to look at in lecture were 2 things. One is affinity maturation. Affinity maturation, and if you look that up, you'll find that is how B cells, over the course of an immune response, make better and better antibodies. And the more you think about this, if you don't know how it works, the more impossible it is, because B cells have randomly generated receptors and they don't know what they're trying to detect, because all they have is randomly generated receptors. So how do they get better and better and better? And that's affinity maturation. The other thing is symptoms of the common cold. Really, I think this is fascinating, and it's way less understood than you might expect. No one can confidently say why you get a sore throat when you have a cold. No one can confidently say why you cough at the end of a cold. Things like that we all experience seem like things we should know. And there are a lot of interesting connections between things like fatigue that you feel if you have COVID or influenza, and fatigue you might feel if you have narcolepsy or chronic fatigue. Some of those are caused by some of the same signaling molecules. And that's all connected to our innate immune response to a virus. So good luck. I'll see you at the next video.